Yeah, I just wanted to say hello and thank you for the introduction and the opportunity to present here today. So I've initially wanted to share our experience with, running the business through the pandemic close downs. But since we had such a successful panel discussions yesterday, I kind of reshuffled my talk. I wanted to talk more about technology to match the purpose of this specific session today. And please, if the audio problems come back, please let me know somehow. We want to start with the notion that everything in our surroundings is kind of run by the laws of physics, and we're all familiar with what the phase diagrams are. With respect to life sciences, the phase diagrams are probably the most complex for water, proteins, and lipids. And again, we all familiar with how temperature affects all of the systems, but pressure has been notoriously ignored mostly for engineering reasons. There were no hardware commercially being produced for controlling high pressure to the extent that actually would make a difference in phase transitions in biological systems. And but yet we have all known from quite a while ago, the work published by a pioneer of the high pressure research at Harvard University, Professor Percy Bridgman, in 1905, I believe, who eventually was awarded a Nobel Prize in Physics for his studies in high pressure, mostly in solid state matter. But he also noticed that high pressure can denature proteins. And at the time, he just mentioned that it might be of some importance in the future. And so this actually experiment we repeated in our systems. And as you see, the level of pressure and the duration of incubation at high levels of pressure actually denatures protein. This is just an egg white that turns solid. So you can essentially cook an egg at room temperature. As a company, we're a small company of about 15 people located in Massachusetts and Boston area. Actually, we have two locations, one north of Boston, one south of Boston. And I'll elaborate on that in a few minutes. We've dedicated to actually using high pressure as a thermodynamic perturbation tool to drive a variety of applications in predominantly life sciences and chemistry. And so we commercialized three platform technologies. Pressure cycling technology, we basically trademark as ability to control pressure up and down from atmospheric level to very high levels of pressure and contain it in our instrumentation to drive biological sample preparation by physical research products. We recently acquired a company called Barafold that developed high pressure protein refolding and disaggregation methods. And so we now continue their mission to bring it into bioprocess development applications. And the third technology platform is we call ultra shear technology. It's essentially a next generation high pressure homogenizer that allows us to make nanometer size emulsions and liposomes. The application areas for the first two platforms cover a wide range of topics and sort of areas related to this specific meeting. We started out in biomarker and target discovery because our past was in proteomics. And then we extended it to the protein characterization and quality control of biopharmaceuticals and biophysical studies of proteins, embracing physics, as we've heard earlier yesterday in talks that drive conformational dynamic studies. And also we could do accelerated stability studies of proteins to develop protein formulations that do not aggregate over time. And as I mentioned before, we also develop protein refolding methods. Why pressure is important? Pressure actually partially unfolds proteins in a very controlled manner. Pressure travels through media with the speed of sound and affects every molecule to the same extent. So there's no diffusion or slow propagation through media as we see with temperature. And so proteins get populated with the solvent molecules, and as a result, they actually swell up or unfold in a controlled manner without addition of thermal or chemical energy. So by using pressure, we can replace chaotropes and detergents to unfold the proteins and keep them that way until the time when we need to for that force to go away. We can just turn the system off and avoid purification with dialysis. We have about 300 products right now in the field in a variety of institutions, the academic labs, pharmaceutical companies, and the government labs, covering essentially this entire circle from target discovery to manufacturing of biopharmaceuticals. And so this is the only slide I left from the topic that matches the title of my talk. As a company supporting all these efforts and the customers who were working on vaccines and therapeutic development, we had to stay open throughout the shutdown. And so as being considered essential, we were lucky enough to have enough business to avoid furloughs or layoffs or any other adverse business decisions. We obviously experienced the plunge in the equipment sales, but as a surprise, we saw the resurgence, or actually the increase in requests for our services, and also consumable sales remained on the same level because of several labs that were furiously working on some of the urgent projects. So support calls slowed down so we could tap into our field support team to repurpose them for R&D efforts. We actually ended up adding a major development project in this homogenizer space I'm going to mention towards the end of my talk. And while R&D efficiency went down due to the fact that we had to stagger our presence in the lab, we've learned the new tricks to be able to actually jump into the lab, set up automation, let's say HPLC runs or high pressure experiments, and then go back home and do the data analysis in the home office such that other people could enter the lab. We saved a ton of money on travel and trade shows. That helped as well. And also being in Boston area, we've experienced a wonderful break from the Boston traffic. We had much easier to find the parking spaces in the city if we needed to move around. And also we had to upgrade all our gadgets, like cameras and microphones, to be more efficient online. So I'm gonna give you several examples of what we do. So obviously I mentioned we started in tissue proteomics because we knew that field and we knew the customers from our previous companies. It turns out that some of this convenient proteases that they used in proteomic sample preparation actually are accelerated by high pressure. And acceleration is bimodal. And first of all, substrate proteins are being unfolded by high pressure, and sort of the cleavage sites are being exposed to the enzyme more readily. But as it turns out, these are experiments done with using the small molecule chromogenic substrates. Some of these enzymes get accelerated when we put them at high pressure, sort of like most of us in the workplace. So essentially what's happening that we can drive the proteolytic digestion to completion to improve the reproducibility and minimize miscleavages when applying pressure and temperature control. This is our flagship instrument called Barocycler 2320XT. It actually has a built-in Windows computer, and it controls temperature and pressure within the wide range of values. This will up to 400 megapascal or roughly 60,000 psi. And there are two versions of the instrument. One is temperature controlled by external circulating water bath, and you can go down to even negative temperatures. Our specs say 4 degrees Celsius just for safety, but you can go down to -20 at high pressure, and your solution will remain liquid, which is an interesting opportunity there. Or the second version of the instrument is heated electrically, and the temperature control is programmed by the computer together with the pressure. The software, GMP version of the software is available for those who require that. We put a lot of efforts in developing sort of CFR 21 Part 11 compliant data storage and safety and security. Part of this instrument is also a consumable, specialized consumable devices that we manufacture. They're made of inert fluoropolymer material, and they have a variety of caps. We call these tubes microtubes, and the variety of caps that allow you to incrementally change the volume, adding reagents and replacing the cap, keeping the sample in the same container. One of the unique features of this consumable device is this built-in homogenizer pestle that essentially works as downs homogenizer, or Potter Elgin rather homogenizer, because it's a Teflon piece. But when customers work with the tissue biopsies, they place the sample at the bottom of the tube, and then by repeated compression and decompression in our system, the walls of the tube collapse, removing the air gap above. And as a result, the sample gets mashed up, sort of like in the manual homogenizer, but this happens in parallel to 16 samples located in the chamber at the same time, very conveniently. You can combine this with the digestion buffer, as it turns out, to further save time and improve the process. So in the conventional proteomic sample prep workflow, typically takes about a day to do the sample prep, overnight digestion, and then the sample is ready for analysis. But when digestion is done in the pericycler and homogenization is done in the same instrument, tissue to peptides workflow could be achieved in a couple of hours. And so as our customers say, the proteomic data is available in clinically relevant time frame. And so most of our customers that work on small tissues are actually trying to push the proteomics into a clinical field by doing either early cancer diagnostics or trying to work on prognostic applications, which I'll mention in a moment. As a result, it's not just a speed, but it's the completion of the digestion process. So we actually see higher sequence coverage, higher total intensity of the peptide peaks, and higher protein and peptide IDs in the proteomic rounds. Several industrial proteomic labs are now resurfacing. And our largest customer in China has 8 instruments feeding a number of mass spectrometers. And they're involved in actually one of the biggest projects this lab is doing at Westlake University is trying to commercialize the mass spec based diagnostics of thyroid cancers that are quite a big problem in China. The next lab is in Sydney, Australia, in the Children's Medical Research Institute. They're basically looking at pediatric cancers, and they have this massive project to sequence 75,000 tumors to try to understand the post-surgical treatments that will be guided by the proteomic content of each tumor, and not by random trial and error. The laboratory at Women's Health Integrated Research Center at Annandale, Virginia, actually is looking at cancer tissue microheterogeneity by combining the laser capture microdissection systems with digestion in our systems, and they actually pioneered this method, after which our partner now, Leica Microsystems, designed a special stage that actually fits our specialized containers right underneath the microscope. So you can collect the tissue sections outlined in the LMD instrument directly into our sample containers, and carry on with the analysis with digestion in the instrument, followed by mass spec. I forgot to mention that pressure also makes some of the ordinary organic solvents chaotropic, so you can skip a lot of reagents in the digestion buffer and basically substitute them with the simple components of the mobile phase. Mass spectrometrists obviously love it because this makes the sample clean enough for direct injection. And so when you deal with several thousand cells in the sample, everything counts. So the fact that we maintain sample in the single container and skip the peptide cleanup allows the groups to actually get the better recovery of the signal and more proteomic coverage. Moving on to protein characterization, basically the same digestion applies to multiple workflows in biopharmaceutical QC. This is an example of the paper published by Genentech Group back in 2016. It turns out that monoclonal antibodies could be digested without being reduced by the trypsinol lyse enzyme, with preserving all of the disulfide. So for the mapping of location of disulfide bond in the product, this is a rapid process, so it takes less than 2 hours. And they see less peptide scrambling, disulfide scrambling, and much bigger yield of the disulfide linked peptides for the product. Another paper that just been submitted and still available as a preprint from our customer at Georgetown University. They actually were looking at the glycosylation of the SARS-CoV-2 spike glycoprotein. And we all have heard the stories about the situations. The glycosylation seemed to affect the recognition by the antibodies and also triggering the proteolytic activation of the virus incorporation into the cells. So this group has actually found several new glycans that are very close to the proteolytic sites. Now moving on to protein refolding. As I mentioned, we purchased a company called Barafold in early 2018, and they've been collecting intellectual property on using high pressure for protein refolding and disaggregation. And most of their work was focused on refolding of proteins from inclusion bodies when they were expressed in bacterial systems. Inclusion bodies are basically precipitated protein, and as it turns out, they're very similar to amyloid fibrils by the way the protein is deposited in inclusion bodies. So they're very rich in anti-parallel beta sheet structures, as shown by FTIR and a few other methods. And so it turns out that both of these structures are very easily disaggregated by just applying high hydrostatic pressure. And so as part of the same instrument, the different mode in the software control allows us to control pressure using the slow ramps up and down. And if you abruptly change the pressure level while the protein is partially unfolded, you're running into a risk of sort of rapid aggregation because protein is ready to interact with its peers at that moment. So by using the slower ramps, we actually eliminate that risk, and this is very useful for pressure refolding applications. This is an example of a textbook case. It's a human proinsulin inclusion bodies before and after pressurization. The 5-minute incubation at pressure actually completely solubilized the pellet of inclusion bodies and in the buffer containing no urea. We actually tried Tris buffer, the higher pH caps buffer at pH 10, Tris buffer at pH 8, and Tris in the presence of DTT. And at higher pH with application of pressure with no urea, we actually can completely solubilize this protein to we haven't tried a higher concentration, but it seems like it's a linear curve all the way to about 13 milligrams per mil. And you see the urea control, it looks very similar to what we can do in caps without in urea. And DTT seemed to be not required in this case because the E. coli has a reducing environment inside the cell, and if the inclusion bodies are fresh enough, all the protein is not cross-linked. And this is how it works. The proinsulin is a single polypeptide chain, and it requires formation of the 3 specific intermolecular disulfides, and then the cleavage of the 2 proteases to remove the C-peptide to form a active insulin hormone. And so we've done, obviously, reverse phase HPLC to show that the unfolded proinsulin monomer actually shifts to the lower retention time area as it folds and becomes less hydrophobic. Also we performed a digestion by the enzymes to show that proinsulin band could actually shift to the active insulin form when it's refolded properly. It's interesting. So interesting. If you add DTT, obviously the A and B chains fall apart, and you get disappearance of the parent insulin band, and the other chain drops down below the front. Now, in the full molar urea, just standard approach, conventional method, the refolding overnight, shows the same exact active insulin, and when the enzymatic digestion is not performed, it's the proinsulin intact band. But when we add urea and a small high pressure method, we don't see the refolding because urea prevents the proper refolding in the presence of pressure. So we can clearly ignore or forget to add urea basically, and save ourselves a lot of time for removing it by dialysis overnight. Moving on to the formulations development, we partnered with the local Massachusetts company called Redshift Bio. They have this infrared spectrometer based on microfluidic chip that allows you to flow your sample through and compare the samples of, let's say, proteins with a different secondary structure. The way we've accomplished this, we developed a chemical environment by adding co-solvent 10% and propanol that allows us to take human IgG fraction and basically create nanoparticles by pressurizing it, partially unfolding it, letting it aggregate to a quantitative, in a quantitative way. And so, as you see, we apply different levels of pressure, 45,000, 50,000, and 60,000 PSI. The infrared spectra show gradual increase in the anti-parallel beta sheet and decrease in the parallel beta sheet, as well as the same relationship is there's some more alpha helixes being formed, and some of the disordered domains disappear. So it's an interesting conversion of the structures that form, somewhat amyloid-like aggregation in solution. The same data presented in a different way. This is actually the 60,000 PSI treated IgG, forms very stable, cloudy, sort of nanoparticle suspension that lasts for several weeks. And it could be used as an analytical standard to quantitatively determine the degree of aggregation by this spectroscopic technique. Curiously, sample that is pressurized without N-propanol remains pretty much intact all the way until about 80,000 PSI when it crashes out. So we believe it's going to be a beginning of a very successful program for us to work together and to form sort of the workflow to quantify protein aggregation in the spectroscopic manner. In order to actually do some high pressure enzymology for our digestion experiment, we had to design a special machine. So it's our pressure generator equipped with the optical cell and the fiber optics-based spectrophotometer. This optical cell has 3 windows, so it supports the absorption, fluorescence, and circular dichroism experiments, depending on what windows we can install. And it fits a conventional spectrophotometer cell with a little cartridge that displays extra water. It tends to fit into most cuvette compartments of commercial spectrophotometers. And so it allows us to do very interesting experiments. What we've also discovered is that some buffers exhibit pH shift when they're pressurized. And this was determined by using this pH indicator Cresol Red. So when you titrate it on the bench with a pH meter, you can actually see the pronounced spectral change as pH changes within the range of between 6 and 8 units. And the same spectral change is observed when you pressurize it from atmospheric pressure to about 420 megapascal. It sort of looks like a traffic light. We actually have a video that shows that the color changes as the pressure goes up in the optical cell. And so to our surprise, well, obviously not surprising, in the water, the pH changes is negligible, whereas some of the buffers like HEPES or phosphate go to almost complete unit when we pressurize it to about 4 kilobar or 400 megapascal. And this is an example of the horseradish peroxidase activity phase diagram, if you will. So the pressure and temperature values and the activity plotted, also obtained with the chromogenic substrate in the optical cell, allows to actually understand the degree of activity impact by applying pressure and temperature. As a result, we have a number of collaborators and customers in top-tier biophysics research labs in the world. These labs are employing high pressure to perturb samples and studied by various physics techniques like NMR, electron paramagnetic resonance, small angle X-ray scattering, time result fluorescence, super resolution microscopy, and some of these techniques are also amenable for pressure jump kinetics experiments. It's very exciting. So moving on to the last part of my talk, nanoemulsions and liposomes. As I mentioned, we now have this new project developing a high pressure homogenizer system. It actually started out as an NIH grant to develop a system to share DNA for Illumina sequencing. and so we developed this microliter scale homogenizer head that was driven by HPLC pump and the autosampler, and allowed us to start with the genomic DNA and obtain sheared fragments of desired size by controlling pressure and flow rate. And then we realized that our design is very flexible and very scalable. So we actually decided to embark on this ambitious goal to develop a production scale or pilot scale system. So the next grant funding came from our collaboration with the Ohio State University with Professor Balasubramanian, who is in the food science department there. And so he obtained the grant from United States Department of Agriculture to actually make an attempt to develop methods to make dairy products shelf-stable, sterile, essentially. And so we designed this concept that allows to combine temperature control, very high pressure, and the high shear forces to completely eliminate any pathogen presence, including the bacterial spores in the product that comes through if the product is pumpable and needs to be homogenized anyway. This is the concept of this particular grant. And so the grant funded the development of a larger system that we use now. This is how the homogenizer valve works. Essentially, most conventional high pressure homogenizers are relying on the fixed size sapphire or diamond orifice, where the product gets squeezed in at a very high flow rate. And so in order to achieve the pressure drop across this orifice, you have to develop very high flow rates. Now, problem with that if you want to increase the efficiency, you have to make the orifice infinitely smaller. At that point, it's prone to clogging. And so we decided to work around this problem. We decided to purposely plug the hole and control the annular orifice that is formed this way by dynamically controlling the forces that counteract the high pressure from the product flow on the other side. And so as a result, we actually maintain this so-called nanogap orifice at several nanometers wide, and the process becomes independent of the product viscosity that comes through. And so as a result, we determined that we could make nanometer size emulsions that are about 50 nanometers in size And it's interesting, they look like true solutions and they behave like true solutions. They're thermodynamically stable. But the only way you can tell that it's not a true solution is by shining, let's say, a laser pointer light through it, and you can actually see the beam. So it actually scatters light to some extent. But since the particles are smaller than the diffraction limit, you don't see them under the microscope when you don't see them floating in solution, like in the conventional emulsions. And so as you see that, as soon as the beam hits the conventional milky emulsion, it basically gets absorbed and doesn't travel through. And this is another example with the pharmaceutical angle. So we basically try to nano-emulsify the prednisone, 1 milligram per mil prednisone formulation in the hope that this nano-emulsions will become more bioavailable, at least more bioaccessible in transcutaneous oral formulation administration forms. And so we're actually currently looking for partners in the pharmaceutical industry to probably help us to jointly develop something like this and follow 505(b)(2) path in the new formulations. And so I just wanted to summarize that high pressure is a very convenient tool, thermodynamic tool for a variety of applications. We have several instruments available to use it in the lab, and we offer them for our customers. We're looking for collaborations. Thank you very much. I obviously have to highlight our collaborators. We are very actively collaborating with academic institutions and industry, and we would not be successful without all of this very, very intelligent people. Thank you very much.
Thank you so much, Alex and Dr. Alex, for your presentation. And there is just one question for you from Hugo, CEO of PhenoSwitch Bioscience. And he's asking you, Dr. Alex, biologics formulation to prevent aggregation is one of the key items during CMC of those. We try to trick the proteins by different means to be stable, at least at injection site. How does this translate once in tissues and plasma?
That's very interesting. So obviously, there are two things that come to mind right away. Obviously, pressure travels through cell walls. And in fact, some of our customers are looking at pressure effects in vivo on bacteria. In fact, they can trigger certain biochemical processes by manipulating conformational changes in proteins inside the living cells. You have to be careful not to go to the high levels of pressure that actually are lethal or damaging. But pressure triggers the stress response in bacterial cells. And there's a whole group of scientists that actually looking at the stress response as opposed to, let's say, the heat shock response. There are some of the proteins that get expressed are transcription factors, and they basically, the SOS response is overlapping, but not exactly the same between the heat shock and the pressure stress. So that's just to say that, yes, pressure could be used to manipulate things inside the compartments like cells and so forth. Now, what we're doing with pressure in the biopharmaceutical space, we're trying to define ways to apply pressure in production, in manufacturing, to actually screen for better chemical environments that prevent proteins from aggregation. Aggregation obviously leads to high immunogenicity and potentially anaphylactic shocks or just elimination of the drug from the patient's system by immune system. And in our equipment, we can actually screen up to 16 different chemical environments to see what would push protein back into its native conformation. In our protein refolding experiments, we actually find that chemical environment is the most critical because pressure inevitably dissociates aggregated proteins. It almost never is the bottleneck in our contract services for protein refolding development. It's the redox when you need to form the disulfides at the right time, and other chemicals that contain protein prevent it from finding its partners and aggregate. And so pressure keeps it from aggregation, but as soon as pressure is gone, the process may start again. So what we're trying to do is use pressure as a perturbation tool to allow it to unfold in a controlled manner and then screen for chemical environment that prevents it.